In epidemiology, we study the frequency and distribution of disease and classify the disease on the basis of level of occurrence as sporadic, endemic, epidemic, or endemic. Sporadic refers to a disease that is infrequently and irregularly present in a given population. That means the cases are very rare but requires an epidemiological investigation. For example, polio, rabies, tetanus. Endemic refers to a constant presence or usual presence of a disease or infectious agent in a population within a geographical area. The amount of a particular disease that is usually present in a community is generally classified as the baseline level. In the absence of intervention and assuming that the level is not high enough to deplete the pool of susceptible people, the disease may continue in a population for an indefinite period of time. This level is often regarded as expected level of a disease. A disease can become epidemic when there is a sudden increase in number of cases of a disease above the baseline level in a given area in a short period of time. For example, dengue. Another term which is used is called an outbreak. It has almost the same meaning, but it is limited to a very small geographical area. An epidemic disease can occur due to an introduction of a more virulent strain of an agent or enhanced mode of transmission or a change in susceptibility of the host response to an agent and other factors. When an epidemic spreads over several countries or continents, it is referred as pandemic. An example is COVID-19. Epidemiologists make use of study design, that is, the manner in which the data are gathered to test a hypothesis, similar to those researchers which study other phenomena in a given population. There are basically two approaches of epidemiological studies, that is, observational and experimental. In observational studies, subjects are not manipulated. However, in experimental studies, subjects are manipulated. Let's discuss both of them in detail. Observational studies are two types, descriptive and analytical. Descriptive epidemiology is a type of observational study in which the distribution of a disease in a population is studied. Relevant information usually include information about the affected people, the place and the period in which the disease occurred. This type of study will involve interviews with the patient, their contacts and their family members. Examination of their samples, medical records and history is also taken. In this type of study, the problem is actually known before the study starts, and this study can also be conducted while the outbreak is still occurring. An excellent example is the cholera investigation by John Snow, which we have already discussed. There are two basic types of descriptive studies, like retrospective and prospective. In retrospective, we use the historical data to identify association with the disease state of a present cases. So it's a backward study, and prospective epidemiological study is a forward study in which we gather the data and follow cases to find association with the future disease states. The second type of observational study is analytical epidemiology, in which we analyze a particular disease to determine its probable cause, reason, and risk factors. It can use different variables to discover possible routes and rate of infection. This study can be carried out in two ways, either by case control or by cohort study. In case control study, the epidemiologist look for factors that might have preceded the disease, in which we make a comparison of two groups, in which one group has a disease is compared with another group which is free of that disease, and try to find out any association between an environmental or genetic factor with that disease. For example, one group with meningitis and one without the disease may be matched by age, sex, socioeconomic status, and location. These stats are compared to determine whether there is some factors which are specifically associated with the group which has disease and it is absent in the control group. In cohort study, the epidemiologist studies two populations. One population had a contact with the agent causing a disease and the other one is which doesn't have any contact with the positive agent or the risk factor. For example, a comparison of one group composed of people who have received blood transfusion and one composed of people who have not received any blood transfusion and with the incidence of hepatitis B virus. This is a type of cohort study. If there is an association found with the blood transfusion with the presence of hepatitis B, then we can say hepatitis B virus is associated or linked with blood transfusion. Experimental epidemiology studies generate strong evidences of causation of a disease or treatment by manipulating the subject and comparing them with the control group. The treatment might be administration of a drug or inclusion or exclusion of different dietary items, physical exercise, or a particular surgical procedure. Generally, the subjects are animals or humans. A clinical trial can be used to test a hypothesis regarding the effectiveness of a drug. A group of infected individuals is selected and divided randomly so that some receive the drug and others receive a placebo or a substance that has no effect. So there are two groups, a control group and a tested group. So clinical trials can be of two types, a single blind or double blind. In single blind trials, the individuals don't know whether they are in the test or the control group, but the physician knew that. However, in the double blind clinical trials, both the subjects and the physician don't know the identity of each group. If all the factors are kept constant between the two groups, and if those people who received the drug recovered more rapidly than those who received the placebo or in the control group, it can be concluded that the drug was experimental factor that made the difference and was successful. So student, it's time for our second concept checkpoint. So you're giving 5 different scenarios, and in each case you have to identify the level of occurrence of a disease, whether it is sporadic, endemic, or epidemic. Try to find out the answers and we will discuss them in our interactive section.